Students, in last video we were talking about entomophilous flowers and their characters. Now let's continue with it.
In some other species, floral rewards, they are in providing safe places to lay eggs. One such example is Amorphophallus, the tallest flower, which is approximately 6 feet in height. It provides safe places to lay egg for the pollinator. For example, moth and yucca plant. Moth and yucca plant, they cannot complete their life cycles without each other. The moth deposits its eggs in the locule of the ovary, and the flower in turn gets pollinated by the moth. The larva of the moth comes out of the eggs as seeds start developing. So there are several other examples of this kind of relationships, which are mutual relationships in which both of the organisms, they get benefit from each other. Next, we are to discuss some outbreeding devices. Outbreeding devices are those devices which the plants develop to avoid self-pollination, because continuous self-pollination leads to inbreeding depression. The monotony is there. So the plants, they develop special mechanisms to avoid self-pollination and to promote and to enhance or stimulate cross-pollination. So we are going to discuss such outbreeding devices which the plants develop. So first of all, in some plants, the pollen release and the stigma receptivity, it is not synchronized. That means they are both the conditions, the two conditions can occur. It may be a protandry condition or protogyny condition. Means protandry condition is that condition in which the anthers mature when the stigma is not ready, and protogyny means gynoecium matures earlier than androecium. So under such circumstances, the plants, the flowers, they are not able to fertilize because one of the partner is not mature properly for fertilization. Second example is that in some species, the anther and stigma, they are placed at different positions so that pollen cannot come in contact with the stigma, and this prevents autogamy. If they are present at different heights, the lengths are different, then they will not be able to transfer the gametes, and fertilization will not be there. Means self-pollination or autogamy is prevented in bisexual flowers. Next important outbreeding device is self-incompatibility. Self-incompatibility is the genetic mechanism. It is the mechanism to prevent self-pollination. In this self-pollination from the same flower fails to fertilize the ovules of the flower by, like, it inhibits the growth of the pollen tube. अपनी ही species का वो fertilize नहीं करेगा on the same flower. So in such cases only cross pollination will be there. Next is the production of unisexual flower. That means the plants, they develop unisexual flowers to avoid self-pollination and to promote cross-pollination. Plants such as maize, castor develop unisexual flowers on the same plant, which prevents autogamy but not dichonogamy. While in some plants, the male and the female flowers are present on different plants. For example, papaya. This prevents both autogamy as well as dichonogamy. Next is the pollen-pistil interaction. This is what happens when pollen grain comes in contact with the pistil. Pistil is the female part, which is when it is mature, it is ready to trap the male gametes. When the male gametes, they are transferred after pollination on the stigma, when they land on stigma, the pollen-pistil interaction takes place. Let's now understand what happens when pollen and pistil comes in contact with each other. Sometimes wrong pollen lands on the stigma, which is not incompatible, which is not able to fertilize on the stigma, or which is not accepted at all. So it is degenerated. If the pollen grain is of right species, right type, only then it will be allowed to germinate. And initially it will be recognized by the stigma and by some factors, and then it will absorb water and swell up. And through the germ pore, it will start forming pollen tube. So the pollen tube, when it comes out from the germ pore, as we know that pollen grains, they are generally shed at 2 or 3 cell stage. So initially it absorbs water and swell up, and then pollen tube carrying the male gametes, it comes out from the generative cell and moves towards the cells of the style. It innervate the cells of style and approach the embryo sac for fertilization. So pollen tube generally carry two male gametes. Two male gametes are formed from the generative cell, and the vegetative cell helps in the formation of pollen tube. So there are different ways by which the pollen tube can enter in the embryo sac. Either it can enter through the thalassal end, or it can enter through the integuments, or it can enter through the, in majority, it enters through the micropylar end. So when the pollen tube enters the embryo sac through micropylar end or micropyle, the important process of double fertilization takes place. So let's now discuss what is double fertilization. It is basically two fertilizations which takes place inside single embryo sac of angiosperms. First of all, one of the male gamete which enters the embryo sac, it fuses with the egg cell and it leads to the formation of diploid zygote. Other male gamete enters the secondary nucleus, or it enters the embryo sac and fuse with the secondary nucleus, which is diploid. And both the processes, they are called as syngamy as well as triple fusion respectively. When one of the male gamete fuses with the egg, it is called as syngamy or generative fertilization, and it leads to the formation of diploid zygote. And in other case, when second male gamete fuses with two polar nuclei or secondary nucleus which is already diploid, the process is called as triple fusion, TRIN, and it is vegetative fertilization and leads to the formation of triploid endosperm. PEN is formed, primary endosperm nucleus. So because these two fertilizations together takes place within one embryo sac, that is syngamy, and triple fusion together referred to as double fertilization. You can see the process occurring in diagrams, figure number 2.12, and next is the 2.13. So let us now discuss the process of artificial hybridization. Artificial hybridization is the main approach. It is the main process, or it is the approach in crop improvement program in which we can produce the desired species by selecting the pollen grain from desired plants. So it is achieved by the important process called as emasculation and bagging techniques. Emasculation is a process in which female part is not disturbed at all, and the male part, that is the anthers, they are removed very carefully from the bisexual flowers, because unisexual flowers, they need not to emasculate. So bisexual flowers, they are emasculated, male part is removed without disturbing the female part, and after that bagging is done. Bagging is done basically by covering the stigma with the butter paper, so that unwanted pollen grain cannot contaminate it. After that we can remove the bag and dust the stigma with desired pollen grains and to produce desired species. So in this way, artificially we can produce we can sprinkle the pollen grain, we can spread the or dust the pollen grains of desired species on the stigma and can produce desired species. We can produce healthy species. So plant breeders employ this technique importantly for the purpose of cross-pollination and to prevent self-pollination, because cross-pollination leads to formation of healthy species, more better adaptable, better adapted species, which lead to evolution. So just go through the notes once. Students, now I hope you understand the process of formation of male gamete in plants and formation of female gamete in plants, and after that now you are familiar with the process of double fertilization. Now we are moving on to various post-fertilization events that takes place in the flowering plants. So post-fertilization events, they include endosperm formation, next embryo formation, and then seed formation. So first of all, we will discuss endosperm formation. Endosperm is defined as basically the food latent tissue which provides nutrition to the developing embryo. This tissue is filled with reserved food materials, and initially it results because as a result of triple fusion, PEN, primary endosperm nucleus, is formed. So this nucleus, this cell undergoes several nuclear division to form free nuclei. This stage is called as free nuclear endosperm. After that, cell wall formation takes place. This type of endosperm is present in coconut. In the coconut, two types of endosperm is present. One is free nuclear, which is liquid endosperm or milk of coconut, and other is a cellular endosperm, which is the white kernel. Because in free nuclear endosperm there is no cell wall formation, but in case of cellular endosperm the cell wall formation takes place, so it becomes solid. Next are the types of seeds on the basis of the presence or absence of endosperm. If the endosperm, if this reserve food material is present in the seeds are called as endospermic or albuminous seeds. Such seeds, they are they include castor, wheat, maize, barley, or coconut. And on the other side, if the seeds, they do not have endosperm, if the endosperm is consumed in the development of the embryo, such seeds are called as non-endospermic or ex-albuminous seeds. And the examples are pea, groundnut, beans, etc. Sometimes in some seeds the remnants of nucellus, they also persist, and this residual persistent nucellus is called as perisperm. Examples are black pepper and beet. Next, we are going to discuss embryo formation in plants. So embryo formation, it actually develops at the micropylar end of embryo sac. Zygote divides after the endosperm is formed, and this assures the nutrition of developing embryo. The early stages of the embryo development, they are same in both monocots as well as in dicots. The zygote gives rise to proembryo and subsequently leads to globular, then heart shape, and then mature embryo, as you can see in the diagrams. First of all, figure number 2.13, you can clearly see globular embryo is formed, then it turns heart-shaped, and then typical mature embryo, dicot or monocot embryo is formed. So these are the different stages of embryo development. Next, we are discussing the structure of dicot embryo. A typical dicot embryo, if you see in diagram in figure number 2.14 part A, typical dicot embryo consists of an embryonal axis and two cotyledons. Cotyledons, they have reserve food material. The portion of the embryo on the axis above the level of the cotyledons is epicotyle, which terminates with the plumule, or stem tip. The cylindrical portion below the level of cotyledons is hypocotyle, that terminates at its lower end. In radicle or root tip, because radicle plumule is the future shoot, and we know radicle is the future root. The root tip is covered with root cap. Next is the structure of monocot embryo. Monocot embryo consists of only one cotyledon. A single cotyledon is there. An example is grasses. And the single cotyledon in grasses is called as scutellum. It is present on one side of the embryonal axis. At its lower end, it has radicle and root cap enclosed in a sheath called coleorhiza. On the above level of scutellum, it has epicotyle, which has a shoot apex enclosed in hollow structure, which is called as epicotyle. Next, there are some important definitions regarding the different types of seeds. So dormancy is the condition, as we have already discussed albuminous and non-albuminous seeds, and we have discussed perisperm. Now we are discussing dormancy. Dormancy is the condition which is characterized by the state of inactivity in which the seeds, they enter during unfavorable conditions. The seeds, they enter in the state of dormancy during the external environmental conditions which are unfavorable for them.